going to be, to start with, how do these CDNTases fit into bacterial pathogenesis? I'm hoping to take a variety of approaches, but starting with a genetic approach, knocking out these genes in diverse pathogens and asking, are they capable of changing pathogenesis in a mouse model? But also, I think that having this set of nucleotide second messengers gives us a unique tool to identify novel nucleotide receptors in the host. At least a portion of those receptors we expect to be for innate immune signaling and for response to bacteria. But I don't think that it's limited to innate immunity, because what I haven't told you about is, in addition to bacterial cGAS-like enzymes, there's a large family of host cGAS-like enzymes that's just beginning to be uncovered in the literature. So as many as 12 to 40 cGAS-like enzymes are encoded by humans and mice. We understand what a portion of them do, but many others probably have dedicated signaling roles, and some of which are in development, others are in immune signaling. And I think we have a unique tool to circumvent understanding those enzymes by adding our molecules to tissue culture and looking at signaling that might be downstream of those mammalian signaling enzymes. With that, I just want to thank the people who helped make this possible. James was an incredible collaborator who helped out with all of the structural side of this work. But in addition, I just want to acknowledge John McNeil and Philip Cranches, who have been very supportive in this project and allowed me to sit really in between a lab that works on bacterial pathogenesis and a biochemistry structural biology lab and take advantage of both of those groups. With that, I can take any questions.
Hi, that was very nice. So having shown that bacteria can make these dinucleotides, can you actually detect them in a mammalian system, say, or even in humans that have sepsis or some sort of immune disease? Can you actually detect the presence of these in a host?
That's exactly the kind of experiments that I want to do going forward. What's interesting about these bacterial cGAS-like enzymes is whereas many of them are on in vitro, we think often they're highly regulated in vivo. And I think that's going to be important for what their biological function is. And I have some hypotheses we can get into. But we're just beginning to understand what are the levels in vivo for these nucleotides. Now, for those other bacterial cyclic dinucleotides, I think that's a place where there's a lot of room to understand what those molecules are doing in terms of the immune response. So cyclic di-AMP and cyclic di-GMP are made by even greater, probably over 90% of bacteria. I think our gut is probably filled with those nucleotides which come from a different set of enzymes, and it would be really interesting to look simply in the bloodstream, what are the levels of cyclic dinucleotides during sepsis?
Jeff.
Great talk. So PAMPs are usually integral physiological features of bacteria in general. So how many of these family members do you find in environmental organisms for which there is little, if any, evidence for host interactions? And what happens when you knock out the gene of Listeria and you knock out the gene in cholera?
Yeah. So what's interesting about these CDNTases is they're firmly in the mobilome. And so they're very different from the other cyclic dinucleotide synthases because although I pointed out in E. coli that has two different CDNTases, in general, only 10% of the E. coli isolates have these enzymes because they're in mobile elements. And so we find them distributed the same way you would expect to find many other mobile elements, where they're often grouped based on the niche that you identify bacteria. So one of my favorite examples of a CDNTase wedge up here is encoded almost exclusively by rhizobia, by bacteria that live within the root nodules of plants. So we find them both in pathogens and non-pathogens. Unfortunately, the sequencing databases are so skewed towards clinical isolates, we haven't been able to do good almost epidemiology to ask, are they enriched in pathogens or not enriched in pathogens? But I think this gets to, there's a core physiological process that these enzymes are participating in, but it has a selective advantage. And it's not, it's perhaps in the mobile specifically because there's only a selective advantage to having this. And so I'll just take your question to jump off and introduce my hypothesis of what these enzymes are doing is it's controlling a form of bacterial cell death. So the DNCV enzyme from Vibrio cholera activates a phospholipase that results in rupture of the bacterial membrane. What I think is going on is DNCV is a sensor for phage infection. And upon phage infection, that elicits a bacteriolysis that limits the reproduction of that phage. There's a lot of data to support that, and a little bit of preliminary experimental data that if you overexpress these enzymes in E. coli, you see protection against T. odd and T. even phage. But that's the direction that I'm going. And I think eukaryotes have evolved to see this core protection from phage and back bacteria into a corner. They can't lose these enzymes because now they'd be susceptible to phage, but when the bacteria retain these enzymes, now the eukaryotic immune system can listen in. And so we've got a lot of room to grow, at least in that direction.
It seems that manganese boosts the activity of the enzyme significantly. Does it change the specificity or cause misincorporation? I mean, did you analyze that?
In a longer form of this talk, I like to zoom in on that manganese versus magnesium TLC. So without going back there, yes, when you add manganese, you overactivate these enzymes. Typically, we don't find that the specificity changes that much, but even a small byproduct is magnified. So it's kind of like turning up the volume on everything. But cGAS-like enzymes are fundamentally polymerase-like enzymes. That fold is a pol beta-like nucleotidyltransferase. And just like in mutagenic PCR, where you dope in manganese to activate and sometimes add misincorporation, we see something like that there. So we focused on only the CDNTases that are active with magnesium, because I think that's physiological. But manganese will turn on some enzymes that are otherwise off in magnesium. And I think there what it says is these enzymes want to make a product, they're well folded, they could potentially synthesize a product, but they're lying in wait for either a stimulus or the proper reactant. We can overcome that with manganese.
But does it alter the nucleophile, the hydroxyl that attacks that?
I don't know. We haven't looked into it enough. Based on the polymerase literature, I would expect that manganese changes the affinity for the nucleotides in the active site, but I'm not sure exactly how the mechanism is working.
That was a great talk. I was wondering, in the eukaryote, the presence of these molecules will now generate these molecules which are considered PAPs, and that would cause an immune reaction. So there must be some way of preventing a self-reaction from happening in the system. So the question is, do you know how that is prevented? And B is, exactly how does this, a molecule like STING, how does that then correlate with toll receptors, for example, in order to activate the rest of it?
I think that's a great question. I always go back to the gut as being this place that is probably filled with cyclic dinucleotides, both these and others. And how are we not, just full of type 1 interferon and responding to those? I don't know. It has to probably do with tissue expression of different receptors. Although STING is well expressed in the gut, it probably has to do also with barrier functions. So these cyclic dinucleotides don't cross barrier membranes freely. But there are a subset of transporters, both in bacteria and in the host now, that can transport these different cyclic dinucleotides. So maybe it's the expression of transporters. But I think it's a great question going forward. How is the immune response regulated against these? What I will say for the bacteria, and in the case of, say, something like Listeria, which secretes cyclic di-AMP directly into the host and turns on an inflammatory response, we find that inflammatory response doesn't usually alter Listeria pathogenesis that much. And my take on that is that Listeria is circumventing whatever the inflammatory response would usually limit a bacterium that was, say, non-pathogenic. And so I think this is going to get back to discrimination of pathogen versus non-pathogen, and probably tissue-specific expression.
And the activation of toll?
And the activation of toll, I don't know. STING, I don't think is altered by pre-treating with toll agonists, but certainly the STING signaling downstream would alter toll signaling.
Right, it would be upstream of toll.